Hello students, welcome to EPG Pathshala. I am Mohan Chand Joshi, Assistant Professor from Jamia Millia Islamia. Today we are going to discuss about a module, Microscope Based Imaging and Application, Part C, under paper Genetic Engineering and Recombinant DNA Technology, Part 1. By the end of this module, students will understand what is fluorescent based microscopy, what are the application of this particular microscopy. There are three major fluorescent based variants of fluorescent imaging. One is wide field, confocal microscopy, as well as two photon microscopy. Also, we will discuss about the few applications that are very well used and very well known in the research area, that is FRAP, FRET and FLIN. And in the end, we'll try to summarize how fluorescent based imaging has enabled researchers to understand or address different biological questions. Fluorescent based microscopy. So the fundamental principle underlying the fluorescent based imaging is that we have a sample which is tagged with a fluorescent molecule which has a luminescence property. That means that particular molecule can be excited when absorbs a specific wavelength and reverts to ground state when the excitation light is removed, and in that process it emits a light with a longer wavelength which can be captured. Now this particular method or this property is exploited by fluorescent microscope, in which we tag our sample with specific luminescent molecules and excite it with a specific wavelength and look at for the signal which we anticipate coming out from the sample. Unlike the light microscopy, where it relies on absorbing that light when it is passes through the specimen, here we are looking at specifically the signal or the wavelength which is emitted by the fluorescent molecules. One of the major advantage of this particular technique is that it is used for imaging the not only imaging to get the spatial localization of the sample, but also use whether the sample molecule or the molecule which we are looking at is diffusing at a specific rate or not, and also we can figure out the interaction between two different molecules, whether they are interacting or not. So what are the core principles that is involved in fluorescence imaging or fluorescent based microscopic imaging? It requires an luminescence molecule which has the capability to absorb energy and also emit energy at a higher wavelength. So for example, in fluorescent based microscopy, molecules which absorbs energy will go to a higher energy state and will remain there and will come back to a lower or the ground state, and during that process it emits lights, and that's the window which is used by fluorescent based microscopy, because it looks at that emitted fluorescent light and captures it, and by that it can also figure out where exactly that molecule is inside the cell or in the specimen one is looking for. Unlike light microscopy, where there's a white light that is used, because in a white light you have all the wavelength that are available, but in the fluorescent based microscopy we further defragment that white light into a specific wavelength that we are interested in, and then try to use that wavelength to excite our molecule of interest and then view it when it emits light when it returns to its ground state. The major advantage of this technique over light microscopy is that first it provides the specificity and also gets very localized. We can target a very specific area which we are interested in, and also it is very fast, accurate in terms of its spatial localization. Also will allow a researcher to understand if a molecule have a diffusion capability, means like if we are spotting a specific molecule at specific location, whether it is diffusing, so we can also figure out what is the diffusion coefficient. So there when a molecule has a Brownian motion, as well as if it is vibrating from one location to another, so we can figure out within that perimeter what is the diffusion coefficient of that particular molecule. And also by labeling two different molecules which we think whether they are interacting, we want to test whether they are interacting or not, and if we know they are already interacting, we can visualize it by tagging two different molecules and then visualize it under the microscope to see that both the signals are coming from very close vicinity. That means those proteins are interacting. So these are few applications that are also done using this method. Here's the schematic that how fluorescent imaging actually works. So as I mentioned earlier, suppose you have a luminescence molecule which is shown in blue here and is in the ground state. Once you have an excitation wavelength which is smaller, which is low wavelength but in high energy, so what happens, your molecules takes up that energy, absorbs it and goes to a higher stage, which is shown in different rows up here. And this is this entire reaction is very quick, as you can see by the time it takes from ground state to the excited state. It remains there and then it comes back to its ground state as the energy is removed. It has absorbed the energy. Now it is coming back, and what happens in this case, that you have loss of energy which results in a higher wavelength or the longer wavelength, and this entire reaction is also very slow. It's very fast, I beg your pardon, it's very fast, and you again see a signal coming out. So the difference between the place the wavelength has moved is also called Stokes shift. That is the point it is got excited to the point it has emitted the wavelength. So that shift is called Stokes shift also. And this entire reaction is very quick. It actually happens in 10 to the power minus 10 to the power 12 seconds, or very quickly. Wide field microscopy or epifluorescent microscopy, or in general it's called fluorescent microscopy. And as the name suggests, wide field microscopy, in this the entire sample, the wide area has been illuminated. And as I mentioned in earlier slides also, the core principle behind fluorescent microscopy is that you have a fluorescent molecule that is tagged with your sample. Now we need to excite that particular molecule to visualize it under a camera or through our eyes. So we need to provide it a wavelength through which it can be excited. Depending upon the fluorophore that we are using, we know what exact wavelength we want. So we have a mercury lamp or the arc lamp which produce the white light, and as you can see in this picture, you have a light source on the left hand side which throws a light, and this light is diverted toward the sample by a dichroic mirror, and this dichroic mirror is coupled with an excitation filter. So that excitation filter will allow passage of only that light which is going to be excite our fluorophore. So by using different combination of excitation filters, we can excite the same sample with different wavelength depending upon the kind of fluorophores and the number of fluorophores that we have in our samples. Thereafter, when the sample is getting illuminated, the entire range, the entire sample is getting illuminated by the specific wavelength of light, what happens, the fluorophore molecules get excited and then emits the light in the longer wavelength, and now this wavelength can come back through the objective lens back to the dichroic mirror, and now there is with the mirror also we have a couple emission filter. Now the emission filter is very specific, again it's a narrow bandwidth emission filter. That means it allows passage of only that wavelength which are longer than our excitation filter, as well as it is a range which we can also select. So in that way we can get very specific and also get rid of any out of focus or any other light which are beyond our known spectra of fluorophore. Thereafter, this entire image can be captured by a camera which is shown on top. So this is the basic principle of wide field microscopy, where the entire sample is illuminated. By using excitation and the emission filter, we can specifically get the localization of our fluorophore. So in the fluorescent microscopy, as I said, that fluorescent molecule is used which has a luminescence property, and then we get different colors or different spectrum from these fluorophore, and depending upon the wavelength range we are looking at, we have perception, we perceive light in different color. For example, the wavelength range from 340 to 400 nanometer is a range which is called ultraviolet UV region. Then 400 to 430 is a violet region where we see the violet light. 430 to 465 is called blue. 465 to 500 is cyan, 500 to 550 is a green color range, 550 to 580 is a yellow color range, and then 580 to 620 is orange, 620 to 700 is red, and 700 onward is infrared. And here is the example of the emission and excitation spectra of a fluorophore. In this case is Alexa 555 that we are representing. So the very first peak you see is representing the absorption spectra profile, and which is shown in with the yellow line. So that means this particular fluorophore Alexa 564 absorbs light with a wavelength of which start from 450 to 600 nanometer, but the absorption peaks at 550. That's why it is called Alexa 4555. But it can be excited and it can absorb light in a very wide range. Similarly, after it reverts back to the ground stage, absorbing the excitation light, it emits the light in a longer wavelength, which ranges from 500 to 700 nanometer, as you can see in the second graph, which is shown in the red line. And the difference, and as you can see, it peaks around 582, and the difference between these two peaks, where there is absorption maxima and the emission maxima, this is called the Stokes shift. So how in the previous graph which we showed, how the excitation and the emission filter that are used to generate a fluorescent image. So here is the configuration which is shown, a filter assembly. So the filter assembly is, as I mentioned in our previous module also, filter assembly contains three important components: dichroic mirror, excitation filter, and the emission filter. And as you can see in the picture on the very right, you can see the light source that is coming, light from the source that is coming is yellow, or the complete light, white light, which has all the spectra, all the wavelength that we want. But it experiences the excitation filter, which removes all other wavelength except the green. In this case, would let it in. Now it will hit the dichroic mirror and will reflect or deflect the light to the specimen. Once the specimen is illuminated with this green light, now the excitation light will come back in the form of emitted light from the sample, which will go back, and dichroic mirror will allow it to go back, and then it will encounter the emission filter. Now the emission filter again will allow specific wavelength to pass through, and that emission light is captured either by the eyes or by the camera. And if it has to shown in this in a graph form, we see that dichroic mirror, as you can see in this case, will allow all the wavelength to get in whichever it encounters. But because the emission spectra, because the excitation spectra, excitation filter only allowed a specific wavelength to get in, and the emission spectra also represent the amount of light that is allowed to get in, is represented in here. So and this is the combination of filter configuration that is used to excite our sample with specific wavelength, as well as to capture the specific wavelength that is emitting out from the sample. As mentioned in the previous module as well, the light source is different. There are different light sources that are available for illuminating the sample, and for the fluorescent microscopy there are a range of these lamps, whether it's mercury lamps, xenon lamps, metal halide lamps, or now the LED light source, which are available and which produce a range of white light, white light with range of wavelengths with different intensity. For example, the very cheapest and the oldest source is mercury arc lamp. That lamp has all the possible wavelength with a different intensity. As you can see, they are very high in terms of intensity. On the other end, xenon lamp has more in have higher intensity of wavelength at more than 800 nanometer, and the most commonly used and most effectively used source for the light source in fluorescent microscope is metal halide lamps, which have relatively constant intensity of signal across the wavelength, as you can see in this. And if it is compared with mercury, metal halide is very comparable. As you can see, the overlay between metal halide and a mercury wavelength. So it's a very most reliable and cheap substitute for the mercury lamp. And in the end, nowadays the availability of LED light source, there are various advantages associated with LED lights. First, as you can see, the intensity for all the LED light source that are the wavelength that are available is relatively very constant. Also, compared to the metal halide, which provides all the spectra in one go, LED can be coupled with filter wheels, which will allow only specific wavelength to strike the sample or to allow to pass through. So in this case, we don't need the excitation filter assembly. Only the emission filter assembly is required, because instead of using the filter assembly on the microscope, we are just having additional filter wheel on the LED light source, which automatically eliminates rest of the undesired wavelength and let only specific wavelength to illuminate or excite the samples. So the fluorescence based microscopy are three of kind. One is wide field microscopy, and as the name suggests, it means a wider area is captured in this. Confocal microscope, on the other hand, has very focused illumination which is guided by a laser, and then also uses a pinhole to get a very sharper image. And third is two photon microscopy, in which just two photons are used to amplify to excite a signal and get an image. One of the application of fluorescence based imaging is FRET, fluorescent resonance energy transfer, and as the name suggests, there is a transfer of energy. And as I mentioned earlier also, that fluorescence based imaging depends upon the excitation of our fluorophore and it returning into the ground stage. And as you can see in this picture, if there is no FRET, then you follow the very similar pattern where you have excitation and the emission. But it is also possible if two molecules are in very close proximity, which is between 1 to 10 nanometer. So what happens in that case, one fluorophore which is excited, and while it is emitting in longer wavelength, and that wavelength can be utilized by the other fluorophore which can get excited in that long wavelength, can excite and also results in a subsequent activation of a fluorophore which can be captured by a microscope. So in this method, what we are trying to understand, and especially that is used to understand whether two molecules are in close proximity or not, and if they are, what will happen in this case, we excite one fluorophore for which we know the kind of wavelength we need, but the other fluorophore is not getting excited by that wavelength. But if they are in close proximity, this fluorophore gets excited and resulting emission wavelength will be utilized by this other fluorophore to get excited and resulting in a fluorescence signal. So this technology has been extensively been used by cell biologists to understand whether two proteins are in close proximity or not, or they are interacting or not. Here is again a pictorial depiction of a standard fluorescent microscope, which is also discussed in previous module. Again, you can see in the you have a reverse L shape frame on which this entire system is kept. We have the light source, which could be mercury lamp, metal halide, or LED light source, through which the light is generated and given it to the dichroic mirror. Before that, it passes through the excitation filter. But in terms of LED, we don't have, as I mentioned in previous slide, in the case of LED, instead of having the excitation filter on the filter assembly, we have excitation filter or filter wheel just after the LED source, which allows only that specific wavelength to get in. Now the light hits the dichroic mirror and then goes and hits the sample. Once the sample is excited and emits that signal, it comes back. Dichroic mirror allows it to pass it back and hits the emission filter, which allows only specific wavelength to come back, and that emitted signal can be visualized by your eyes through eyepiece. or can be taken to the camera for capturing the image. Confocal microscopy. So we discussed about the wide field microscopy where the entire sample is illuminated. In compared to that, in confocal microscopy, instead of entire sample, confocal only illuminates with a specific wavelength to very smaller section of the sample. As you can see in this picture, on the focal plane, the laser is used, which hits the dichroic mirror, and via objective lens, it only illuminates a very specific section that we are looking at, and thereby it removes all the autofluorescence or the background signal that is coming from the sample is reduced. Thereafter, the signal which is emitted from the sample is going back through a pinhole. Now this pinhole further removes any out of focus light as well as any autofluorescence that is coming from the sample, and which is captured by a camera or seen under through the naked eye on a computer screen. We get a very sharper image and a better image. So the image will have a better lateral as well as a vertical resolution in confocal compared to the wide field microscopy. And here, as you can see in this schematic, again we're showing how the point spread function or the specimen illumination differs in both the cases. In wide field microscopy, we are illuminating a very large section. As you can see, the specimen kept is a glass slip, but a wider area is illuminated compared to the very specific point in the lasers of confocal microscopy. And the resulting image from both of these microscopies are like this. The same three images are viewed by wide field microscopy as well as confocal microscopy, and as you can see in the very first panel, all the three, you see that the images are quality is good, but the resolution is not that great. You still see a blurry image in all the three cases, but in confocal we see a better resolution and much more clarity in terms of the organization that we are seeing in all the cases. Therefore, confocal provides a better resolution, but in wide field microscopy nowadays is coupled with deconvolution method. So what happens in this case, all the out of focus light which is creating this blur image in the wide field is removed, and the sample is concentrated, or the signal is concentrated back to its point of origin, and we get a quality which is equivalent to the confocal microscopy. Two photon microscopy. In two photon microscopy, as instead of fluorophore getting excited with a range of, or the bombarded with the large number of photon, here the fluorophore is excited using only two low energy photons, and photons are concentrated in a specific region so that we have higher specificity and the low background signal. In this case, the major advantages associated with this microscopy is that as the longer wavelength is used for excitation, it gives us a better resolution, it gives us a better penetration, particularly samples which are thicker, and thereby we get a better resolution in vertical resolutions. The number of photons that are used for excitations are also very low, which means there is a low toxicity, low photo bleaching, and sample is not getting affected, and we can image it for a longer period of time. Third and most important is that the background signal is very low, because number of photons that are used for excitations are not a huge number. Therefore, the likely of getting background signal is very low. And third, it does not require a pinhole to image it back. As simple as because a sample is excited in a specific plane, so we know where the signal is coming from, so we can actually track it. It's special and localization. That's why we get the higher resolution. So two photon microscopy provides a very good resolution compared to wide field and confocal. However, it is important that the fluorophore that we are using has a better intensity and has a higher intensity and has a heavy duty cycle. In a sense, it can be imaged for a longer duration of time as well. Here is the schematic of how a two photon based microscopy is used, and if you remember the earlier picture which we showed, where we have a single where we have our sample is getting excited and reaching from ground state to excited state in one go. But in this case we use low energy wavelength or the longer wavelength photons to excite it, and each photon gives half of the energy that is required for it to move from ground stage to the excited stage. And this stepwise excitation is done by using the laser, and again once it reaches to the excited state it comes back to ground state emitting the longer wavelength. The rest of the principle is same, but here as you can see in this specimen also showing, we have specimen is thicker, focal plane can be way below focal plane, and the signal distribution can be separated by in a thick sample. But because we know where exactly we are getting we are exciting, so we can exactly figure out where the signal is coming from. Total internal reflection fluorescent microscopy. Now this is the latest and the high-end microscopy which is used to generate a high resolution image, and this is particularly used for the imaging of the membranes. The principle it exploits is based upon this norm which we discussed in previous module, and so actually it's again creates the evanescence wave, which are nothing but the electromagnetic wave, but they do not travel. So within that area, within the sample, if the evanescence wave is generated, we get the light which are and then we get the signal coming from the sample depending upon its interaction with the waves, and would help us to generate a better quality image. So the excitation beam of the light from the laser is sent to the specimen through a very high objective lens. They also use prism, but prism has several disadvantages. But once the light is sent through the objective lens, it is sent through a specific angle, so resulting in a which results in a total internal reflection or the evanescence wave that is created between sample and the cover slip. And the longer it stays in there, it gives us better information about the sample structure. One of the major advantage of turf is that it exhibits very high resolution and very high signal to noise ratio, and signal to noise ratio is very important in microscopy, as represents that compared to the background how much actual signal that we are getting. So here is example of it. In a wide field microscopy, the signal to noise ratio is almost one fold higher, 1.3 to 2, while in the turf microscopy we can get a signal to noise ratio up to 3.5. And as you can see, the background versus the signal, and the darker background with a whiter signal means that you have lower background and more signal. That's why you have higher signal to noise ratio compared to the wide field microscopy, where we have a very bright image and also have a brighter dot in it that represents the low SN, low signal to noise ratio. And as I mentioned earlier also, turf has an application in for thicker sample, where we want to see we want to visualize endocytosis, exocytosis, as well as looking at the trafficking of protein within the cells. Also it has application in determining the interaction between cells and different kind of cells. Application of fluorescent microscopy. One of the there are few advantages and a few applications which are very commonly used for microscopy. Fluorescent based microscopies are FRET, which is fluorescent resonance energy transfer, FLIM, fluorescent lifetime imaging microscopy, as well as the FRAP, which is fluorescent recovery after photo bleaching method. FRET. So FRET is a method, as I showed in the previous slide as well, that in which one fluorophore acts as a donor while the other fluorophore acts as a receptor, which is important to identify whether two molecules are in close proximity. That's why they can transfer and get the signal from the other fluorophore. And the crucial fact is that it requires both of them should be in very close proximity, almost Forrester radius, that is 1 to 10 nanometer, and for the transfer to be very efficient, the acceptor and donor should be in same orientation, and the application of FRET is in receptor ligand interaction. They are also used in nucleic acid hybridization, as well as in looking at the transport of lipid across the membrane or within the cell. Fluorescence lifetime imaging microscopy, FLIM. This application is based upon the delay that we see in the excitation and emission of the fluorophore, and each fluorophore has a unique natural life that can be manipulated that actually get affected by the cytological conditions such as ion concentration, pH, oxygen concentration within the cell, and protein-protein interaction. So this particular capability or the potential of fluorophore is exploited in terms of understanding how the proteins are moving and dynamic within the cells. For example, RAS, CAM, and RAS in neurons, as well as it is used in multiphoton tomography to detect intradermal cancer also. The fluorescent recovery after photobleaching is a method which is commonly used to understand how quickly the signal is restored after the photobleaching. This particular method is used to understand how efficiently the protein is getting folded within the cell, and also it has been used to understand how quickly a site is being getting repaired during the once the DNA damage is encountered. So a DNA binding protein which binds to the DNA damage site are tagged with fluorescent tag. So once the DNA damage is induced, the protein tagged with the fluorophore will come and recruit in that place. When the repair is over, the fluorescent signal will disappear. So this method is used to determine the dynamics of how protein is getting recruited and getting abolished from the place. So in this picture, as you can see, and in the slide B, that area was bleached, and in the section C and in section D, we see the complete recovery of the signal. So this can be used to understand the dynamics of protein folding and its protein folding and its restoration as well. So by the end of this module, I hope students have learnt about the fluorescent based imaging methods and the microscopy that has been done using fluorescent based fluorophores, wide field microscopy, confocal microscopy, two photon microscopy, as well as the applications of these techniques in FRET, FRAP, as well as in TURF. So in wide field, as I also already mentioned, that wide field and confocal differs in terms the way the sample is exposed. In wide field, the entire sample is being exposed to the fluorescent light, so the entire sample is illuminated. On the other hand, in confocal, we have very precise activation of fluorophore in a specific location, and also by using the pinhole, we are also getting rid of any background light. So compared to the wide field, confocal produces a very high resolution image. Two photon, on the other hand, is a very technique which allows much more specific as well as higher resolution, because in this the fluorophore is excited with just two photons, and in combination of these ones they produce a better resolution compared to confocal and wide field microscopy. And this is very important in terms of visualizing samples which are thicker and your signal is localized within that thick sample, so you have better deep tissue imaging in this. Also, the application of FRET, which is very important in terms of understanding whether two molecules are in very close proximity, because one used as a donor, another one as an acceptor based upon their excitation and emission spectra. Another one is FRAP. FRAP is a technique which is very well used nowadays to understand how the protein is getting assembled, what is the dynamics of that particular to get refolded again, and also is used in understanding how DNA is getting repaired. Also, there is one of the application of this technology is to understand how quickly a DNA damage is repaired by looking at the appearance and the disappearance of a specific protein which are tagged with a fluorescent protein. And TURF is the is a very crucial technique which has been used to understand the membrane dynamics, and which is based upon evanescence waves system, and it allows researcher to better understand how protein interaction that is happening on the membrane, as well as the overall structure of the membrane. So fluorescent microscopy has a range of applications and range of capabilities that allows researchers to understand or address specific biological questions. Thank you.